Thank you so much. Is the microphone on?
Yes.
Thank you, Naveen, for a very generous introduction. In fact, I don't think my mother could have done better. In fact, I don't think my mother would have done better because she knows me. So when I was a beginning student, one of my professors told me that the first rule of a good presentation is that if you outnumber your audience, you should stop talking. So I want to thank you for all being here so that I can give this presentation. And I want to thank everyone here. This has been, as it says, an enrichment experience. It's been a remarkable enrichment experience for me, and an honor to be among such distinguished speakers and such fine faculty and so many enthusiastic young scholars. So thank you for this opportunity. I'd like to tell you today about stuff we're working on, I hope, even in my absence. Someone asked me this morning if these were, is there a thing? This is the first organizational glitch I have seen. This is astonishing. I'm going to do it digitally. No, so we're going to...
So you have to use, I think, just this.
Yeah, where's the whole, where's the thing?
So you just...
The pointer is here. This is a pointer.
This looks like a mouse.
Yeah, because this is a screen. You cannot point a pointer. Right? Yes. You cannot have a pointer. Sorry.
I have a pointer. Okay. So I'm going to tell you about some work that we're doing in my lab. And the university, public universities require me to say that we have a startup company and that if everything works out, I may benefit a little bit and they'll benefit a lot. I don't think there are any spies here, but I need to say it just in case. So what I'm going to talk about is antibiotics. And the CDC, or the Center for Disease Prevention and Control in the United States, issued a report in November in which I've summarized 119 pages here on one slide. And what it says is the antibiotic crisis is here, it's now, and it's affecting everybody. One of the reasons for this, of course, is the decline in new chemical entities, or NCEs, compounds with different structural types that can be used as antibiotics. And I think the decline here is quite obvious. So there are obvious strategies for improving the odds for humans. One way could be if we can increase the penetration of antibiotics into bacterial cells and not into human cells. And another issue that's very important to us, and I hope I can convince you is important generally, is that cells require a balance of cations and anions to survive. These concentrations of ions are very closely regulated by a variety of pathways. So if we can disrupt that, we can weaken or kill the cell, the bacterial cell, but of course we don't want to kill the human cell. And this is sometimes called the therapeutic index. And I can give you an idea of this. Say you're having a party in your backyard and a mosquito lands on your friend's head. You go over with a sledgehammer and kill the mosquito. Now, you probably will kill the mosquito and you'll lose a friend. But if you use a fly swatter, you'll kill the mosquito and your friend will just say, ooh. And that's therapeutic index. You need to be able to kill the bug and not the person. So how do we go about doing those things? Well, bilayer membranes bound all cells, and they're not generally as orderly as this, but they are a barrier. And the way that nature gets things through, even though it's built this barrier, is to use molecules that are either carriers or channels. And by far, the channels are more common. So a carrier is very much like a ferry boat. It picks up something on one side of the membrane, carries its cargo across, and dumps it off, and then goes back for more. A channel, on the other hand, is like a tunnel. And many cars can go through, and they can go through rapidly, but there can only be, say, one or two at a time. So there are limitations there as well. This is a supercomputer model of a membrane, one that I like very much, except I've never been able to figure out how these two leaflets of the bilayer actually stay together. And I don't know if in these calculations there's a parameter for love or gravity or something else, because it looks like they're not in contact. Anyway, the way a carrier works is the carrier grabs the cation and then slowly diffuses across the barrier and deposits on the other side. A channel, on the other hand, has a number of cations on its surface, forms a pore, and then these travel through much more rapidly. And the channels that do this have been recognized for more than 150 years, but the structure of the first channel didn't appear until 1998, when McKinnon published the structure of KCSA, which is shown here in two different views. And I think you can see that these are relatively complex molecules. So we wanted to try to emulate this, see if we could make a synthetic channel, and we were generally discouraged. People said, no, I don't think you can do that. That's a silly idea, especially some of the reviewers of our first proposals. Notwithstanding, we need an amphiphilic structure, and it must insert into the bilayer and create a pore within it. In addition, the structure should have, or it should induce, some kind of a pathway so that ions can flow through. And it would be ideal if it had a selectivity filter. That is, it passed some ions, but not every kind of ion. And finally, because we have coworkers, we need to have a modular synthesis of this so that they can all get their PhDs in less than a lifetime. So those were the parameters when we started. And we have since made a number of families of molecules. I will be talking mostly about these molecules known as hydrophiles. And these are cation selective. These are anion selective, and this stands for synthetic anion transporters. And these heptapeptides are very selective for chloride ion. This was our effort to make an evolutionarily simple molecule and see if some very simple structure would actually exhibit any kind of selectivity, because in the earliest days, there must have been very simple molecules that functioned as channels. And remarkably, this silly little structure shows both cation and anion selectivity. Okay, we call these molecules hydrophiles, and we call this one a benzyl hydrophile because the termini are benzyl groups. And these are related to a carrier molecule that we developed many years ago called lariat ethers. And these molecules initially had donor groups like oxygen here that helped the complexation. The ones we'll talk about today are only alkyl chained. And we want to use these molecules as controls because they're very similar in structure to the central part of the hydrophiles, but they function by a different mechanism. So our initial design strategy was to use two crown ethers as the entry and exit portals that would be on opposite sides of the bilayer membrane. There would be spacers that would define the overall length of the structure. There would be a central unit or a central relay, about which I'll speak a little bit more later, and then side arms that could fold back and fill the void when this went into the bilayer and created a defect. And our initial idea was that there would be a central macrocycle and the ions would go through it. Now, the idea was that that would lower the energy of the transport in the least polar part of the membrane, the midplane of the bilayer. And I've put here two arrows in opposite directions that indicate this is non-rectifying. That means the ions can flow with equal probability in either direction. That idea turned out to be close, but not correct. and I'll show you a little bit of evidence in a minute. But this structure here is actually a hydration point where the energy is lowered by exchanging solvation on the cations that are passing through. And in retrospect, it seems silly that an ion would want to go through another impediment in the center. Nature always takes the lowest pathway. And in fact, this is how the structure actually functions. In fact, when we saw the structure that came out of McKinnon's structure, it turns out that their molecule has openings at either end, and it has what's called the water and ion-filled pore in capsule in the center of the membrane, which is a hydration transfer. So we made some good, perhaps lucky, but some good guesses. Now, we did a lot of biophysics, and more than a year of work is summarized on this slide. We know the membrane is about 35 angstroms wide. We know that these fluorescent dancyl residues are spaced about 28 angstroms apart. We know that from fluorescence resonance energy transfer that the molecule functions unimolecularly. The actual experimental number is 1.1 from the experiment, but that's pretty close to 1 and not to 2. And so we know that it's actually in the membrane. The dielectric encountered by this dancyl group is about 25, and that is actually the dielectric of the glyceryl portion of a bilayer. The inside of the membrane is going to be 2, 3, 4, 5, 6 dielectric, and the outside is going to be in contact with water, so it'll be about 80. So the real action chemically is near that glyceryl area. Now, I said that these were non-rectifying, and this molecule, which has C12 sidearms, was the first compound we made. It was made by an extremely talented Japanese professor who was on my doing a sabbatical with me, and he said this is the first channel, and he called it channel 1, and so it's always been channel 1 in my lab. But the idea here is that ions can go in either direction, and that's non-rectifying, and that's going to be key to what we're going to talk about next. So I said that the Lariat ether was a synthetic ion carrier, and we wanted to use that as controls in some of our experiments. And we will do that in a moment. This is how we envision the channel works as a pore, as a unimolecular pore inside a membrane. There is undoubtedly a column there you are, a column of water and ions. How they're organized, nobody knows. You can't do a crystal structure inside a membrane like this in a dynamic sense. But it's got to be something like this. Now, the profile of transport when you have a carrier is like this. You have 100% of your ions on one side, none on the other. Your ferry boat or carrier carries these across until you have 50-50. And transport continues, but of course there's no change because in the concentrations, because there's no longer a gradient at 50-50. But this is the experimental profile you observe for a carrier. The experimental profile that you observe for a channel is this, where you have a baseline when the channel is closed, or if there's no channel, you simply have a baseline with no transport. When the channel opens, then you have fairly consistent openings because only a certain number of ions can go through the channel at a time. So the experimental profiles that we observe are very different for these. And for this compound that is the C12 benzyl channel, C12 meaning that the spacers are dodecils, I show you about 20 or so transitions here on this slide. But as a matter of fact, in this experiment, we saw more than 30,000 transitions over a 4-hour period. So this is an extremely robust synthetic channel. We used the dancyl channel for our fluorescence biophysical studies, but we wanted to see if we could get evidence for insertion in an actual organism. And so we did some photomicroscopy on E. coli. So this is an E. coli bacterium, which is about 2 or 2.5 microns across. And I think you can see that the concentration of fluorescence is here in the boundary layer of this gram-negative bacterium. If we're disrupting ion homeostasis, that is, we're disrupting ion balance, then we would expect bacteria to be adversely affected. So what we did was what's called a Kirby-Bauer experiment. We take a plate with nutrients on it, and we put a cellulose disc, which is a round piece of filter paper, and you dip the filter paper in various things, put it on the top, let the bacteria grow. If they don't grow, as is the case here, you observe what's called a halo, and that halo indicates that you have antibacterial activity. And this is the result for the C12 channel shown at the right. Now, what we did in this case for a control was use a channel that was too short to span a bilayer and didn't show very good transport behavior. So we compared those two in a quantitative way, and if we call this entry, this halo, 100, this is about 15. Why would there be any from something that's not a good transporter? Well, because it can also function as a carrier, and if it's a carrier, it can also disrupt ion balance. But it is obviously not nearly as good. In order to determine the exact potency of compounds against any bacterium, we do a study that's called minimum inhibitory concentration, which many of you may already know. And the idea is you put bacteria into a test tube, and then you add an antibiotic, and then you add half of the antibiotic and a quarter of the antibiotic and a 16th of the antibiotic, and so on. And when you get to a place where there's no bacterial growth compared to there is bacterial growth, then this is defined as the minimum inhibitory concentration. And this is usually expressed in the medical community as micrograms per milliliter, but we use micromolar and nanomolar, which is also used in the medical community, but not as much. So here it would be 6 micromolar would be the minimum inhibitory concentration for this drug. And here we compare a number of compounds where these chains here are varied in length. And what we see is that for the C12, 14, and 16, against gram-negative E. coli, gram-positive Bacillus subtilis, and the primary eukaryote, the fungus Saccharomyces cerevisiae, we have pretty good activity for these compounds as drugs per se. That means using this instead of, say, penicillin. And you can see that here against Bacillus, we're at about 250 nanomolar. So that's actually a very good concentration, a good efficacy. And if we compare the chain lengths with the killing, we see that the best killing occurs when the chain lengths are appropriate for the thickness of a bilayer, between 12 and 14 and 16. And in fact, if we measure the transportability using those same parameters, we see that there's an inverse relationship, which makes perfect sense because the more we disrupt the ion balance, the more efficacy we should see in killing bacteria. Now, we know that the hydrophiles insert into the boundary membranes of the bacteria and yeast. We know that they're non-rectifying and so that they can disrupt ion homeostasis. We know that certain of the hydrophiles are active at low micromolar or reasonable nanomolar concentrations. But generally speaking, they are also toxic to humans at the concentrations at which they affect bacteria. So that's this therapeutic index problem. Now, we've looked at, when I say we have looked at, the NIH in the United States has what they call a preclinical services program. And if they think you have any prospect in your compounds, they're willing to do testing for you. And they've done hundreds of tests for us, which we sincerely appreciate. These are probably the worst of the existing organisms at the moment, and they're generally referred to as the escape pathogens because of the first letters of the organism's names. So we looked at many of these, and I've just shown a few, and in fact, I just want to focus attention on 3 of them. Enterococcus faecalis, Acinetobacter baumannii, and these abbreviations here, this means the vancomycin, the strain A that's resistant to vancomycin. This means the wild type, or the one that normally occurs, and this means the fluoroquinolone resistant strain of Acinetobacter. And I think you can see we have pretty good activity here against these horrible organisms. So what we think is happening is that when we form this defect to form a channel, we also are allowing greater penetration of the antibiotic. And that's illustrated schematically here. But we know there's justification for thinking that, because colistin, or polymyxin E, which is shown here, is a drug that is known to increase membrane permeability. It's been approved for about 50 years, but it's used as a drug of last resort. So that means if you have a cut on your hand, you don't use this because it has toxicity. But if you're dying of sepsis, you can afford a little bit of toxicity, and this was used in really dire cases. Unfortunately, from 2017 onwards, there have been a number of reports of resistance development to it, so it's no longer a drug of last resort. Now, we wanted to see if the presence of our hydrophiles could, because of this increased penetration, enhance the potency of drugs. So we chose 4 FDA-approved drugs, well-known ones, that have substantially different structures and for which their mechanisms of action are known and different. The mechanisms of action all have something to do with peptide synthesis, but they all function in different ways. And I think even if you're not an organic chemist, you can see the structures are extremely different. And these are different classes of antimicrobials. So we used channel 1, which has alkyl side arms, C12 side arms, and our best results are typically with these benzyl groups, which appear to form cation pi interactions with the head groups of membranes, and that stabilizes them. But that's in another entire story. So we're going to compare the efficacy of these things. And the way we do this co-administration experiment is we determine the MIC for the antibiotic against the organism in the absence of anything else. Then we determine the MIC for the hydrophile in the absence of the antibiotic. And then we add the hydrophile at 1/2 its potency, and in the hope that that is not a toxic level, and I'll show you later that it is not a toxic level. And then we determine the MIC for the combination of the two. And if the MIC is lower, if it's gone down, we've increased the potency by the presence of the combination. And that is usually referred to as synergy, additivity, cooperativity, and the terms are often used interchangeably in the literature. And I read a book about synergy, and I wasn't entirely sure at the end what I had learned. So these are the results of this early experiment. We used 2 drugs, rifampicin, sometimes called rifampin, and erythromycin. Now, erythromycin is a lousy drug against E. coli. It would never be prescribed for it, but it gives us a big dynamic range to work with. So that's one reason we chose it. But I think you can see that by adding the hydrophile, shown here, this is the C12, the C16, or the C14 benzyl, or channel 1, which has alkyl here instead of benzyl. I think you can see the potency of rifampicin is dramatically increased, and even erythromycin shows increases, although less dramatically so. But it's a good test substance for us. So we wanted to know if the synergy was really just some combination of toxicity effects, or whether the hydrophile somehow, even at half concentration, weakens the bacterium. So we did what's called a growth curve, and what that means is we used our half MIC concentration of the hydrophile, which is 1 micromolar, and we grew E. coli, and you can see that there's really no difference in the growth of the E. coli. So that's very clear evidence that it's not a toxic effect. Now we add our erythromycin, 25 micromolar. That's 1/8 the concentration that actually kills E. coli, and we see some decrease in growth. We double the amount, double the concentration. We see approximately the same increment now. But when we add 1 micromole, half concentration of hydrophile, and the low concentration of erythromycin, now we have a very potent combination. Does the hydrophile disrupt the membrane, or does the fact that we're disrupting ion homeostasis affect the efflux pumps so the antibiotics can get in, but they're not readily pushed out? Or do both of those things happen? This is a rewording of that scientific giant called Google. I heard of it before, Google. I just want to point out, this is a quote from them: "Efflux is important in medicine as it can contribute to bacterial antibiotic resistance." And that's certainly true, but the issue is that an efflux pump will pump out any exogenous material, and some of them are actually in resistance, are now selective for particular antibiotics, as you'll see in a moment. So the way these things work is these are big, long proteins, and there's part is in the cytosol and part is above the membrane in the periplasm, and the exogenous material is grabbed, and there's a conformational change, and it's pushed out. And that conformational change is energy dependent, and energy dependence is dependent on ion balance. So we decided we'd make a resistant strain. So we used a competent strain, for those of you who know about these things, JM109. We transformed it with the TET-A efflux pump plasmid, and we call our new E. coli TET-R because it's resistant to tetracycline. The TET-A efflux pump pumps out selectively tetracycline and is highly resistant. Instead of being somewhere around 10 or 15 micromolar, it's now 1,000 micromolar. That's 1 millimolar, which is an absurd value. So if we look at, we were using the strain of E. coli called DH5 alpha for this test. That has a MIC with tetracycline of 12 rather than 1,000. So we have very high resistance in this strain. And it's very selective. You can see here the close relative of tetracycline is minocycline, which differs only a little bit in structure, and the MICs differ profoundly. So we take this compound, which has this 1,000 micromolar MIC. We add half our concentration, half the potency of the hydrophile, so that it's not going to have an effect on the growth of the bacterium. And then we add tetracycline, and we determine the new MIC. And what we find is we now have a MIC of 30 rather than 1,000, which means that we've enhanced the potency by 33-fold. And this is, for us at least, it was a stunning result and a very gratifying one. Now, we wanted to do some control experiments, and we thought lariat ethers would be perfect because we know that those compounds are carriers rather than channels, and they also are this fragment of the molecule, so in a sense they're the perfect control, except they weren't. So we did the same experiment. We used our TET-R organism. We used half MIC concentration of the lariat. Then we added, in this case, tetracycline again, and now we have an 11-fold increase in potency, which is lower, but still very significant. So what can be going on here? Well, we did the same experiment with rifampicin and found that we got quite a significant increase here as well with the lariat ethers. Now, I apologize for these partial structures, but when I was getting ready to give this talk, Professor Kim came in and he said he needed some more nitrogens and oxygens for his talk. And so I gave him mine. So that's why these are missing here. But you can see that the MIC value goes from 60 to 6. And this is a 10-fold increase. So this really was disconcerting for us. It didn't seem reasonable. We've looked at a bunch of organisms and a bunch of microbes, and it always happens. So our basic idea seemed simply to be contradicted. But we did a study called Planar Bilayer, and I showed you what the characteristics of a channel are, the so-called open-close behavior. And when you do a planar bilayer voltage clamp study, you get a trace that would either be that kind of carrier curve I showed you, or the open-close behavior that's characteristic of channels. And this is what we found for this molecule, which has either C8 chains here or C11 chains here. And whether we applied a positive or a negative voltage, we saw that we had significant channel-like activity. And one thing that's a characteristic here that would strike anybody who's familiar with these is that it looks like we have multiple states. And indeed we do. And the way we understand this is that the Lariat ethers are inserting into the bilayer and aggregating within the leaflet, the upper leaflet. We know that when channels are not long enough, the lower leaflets will open up to allow, and there'll be a condensing of the bilayer. And so we know that we have, from a lot of deconvolution of those traces you saw, we know that we have either 5 or 4 Lariat ethers forming a pore, and the ion goes through the center of the pore, not through the center of the macrocycle. So these compounds actually are potent in their own right, and we have some data that suggests they can be extremely interesting as well. So we did a growth experiment with the Lariat ether. Again, E. coli is not affected by a half Mc concentration. When we add tetracycline, we see slow growth. Eventually, we get up to the same point. But when we have the combination of Lariat ether and tetracycline, we see extended potency. Now, when we did this study with I think this is the C11 compound, you can count for me. If it's an additivity issue, we expect to see the data points fall on this line. But what we see here is that we get a much larger increase in potency, much faster than expected, and that's a definition of a synergistic effect. So we're seeing synergy with these compounds. Now, we wanted to try to visualize what was going on in the cell, and so we used these two molecules. This we refer to as FDA, it's fluorescein diacetate, and when it's inside a metabolizing and healthy cell, endogenous esterases or amidases will cleave this acetate group and produce the well-known fluorescent dye fluorescein, which is bright green. So when we study an organism, if it's fluorescing green, we know it's alive and doing well. Propydium iodide does not go through bilayer membranes because of all these charges, and that's very restrictive in bilayers. So if it does get into a cell, it will intercalate into DNA and give a bright red fluorescent signal. So we did a study with human embryonic kidney cells, which is a mammalian cell, and you can see that there's very little penetration of propydium iodide, but it's very healthy in metabolizing. If we use the detergent Triton X, we kill the cell, and all of this, oh, there we are, and all of this is just the detritus from the ruptured cells interacting between DNA and propydium iodide. Now, these are the hydrophiles, and I think you can see that they're all of the organisms are functioning, and here you can see that we're getting significant penetration of propydium iodide. So we know that we are getting enhanced penetration. A colleague, former student of mine, is a professor at another university, and said he would do a very preliminary animal test. We haven't published this because it hasn't been reproduced. But it's interesting, and I think shows the value of the hydrophiles as drugs per se, rather than as adjuvants. And we used fluoroquinolone that's called norfloxacin as this substitution pattern. And so this is not a combination study, I want to make that clear. And we used Staph aureus that had a fluorescent tag, and you can see that we gave the mice, this is a thigh model study. We gave the mice an infection, and then we added norfloxacin or the macrocycle, and at 24 hours, I think you can see we were doing a lot better with the hydrophile. Now, I asked Matt if he would test the toxicity, and he did this on nude mice. He gave them a 10 times dose, which we expected would kill them. He said that they turned bright red within a couple of hours, and the next day they were normal and lived normal lives. So whatever toxicity they encountered was apparently transient, although the disclaimer, of course, is that we didn't do a necropsy. So we know that these form some kind of a pore or defect in the bilayer. We think that gets more more antibiotic into the organism. And we also think that efflux pumps are being blocked, which would keep the antibiotic from being pumped out. So what evidence do we have for that? Well, this has got a lot of stuff on it, but I want to call your attention only to 2 lines. This is inhibition of efflux pump by our C14 hydrophile, and I want you to note that it's at a concentration of 4 micromolar. The standard efflux pump inhibitor is reserpine, which is usually reported at 25 micrograms per milliliter, but 25 micrograms per milliliter is 40 micromolar. So we have activity at 4 micromolar that's much higher than at 40 micromolar for the standard. So there's no question that we're getting efflux pump inhibition. In fact, just this a couple of months ago, we finally had this patent allowed, which claims the use of these compounds as efflux pump inhibitors. And of course, all patents have this kind of very catchy title. Molecules that inhibit efflux pumps in multi-drug resistant bacteria and uses thereof. But of course, the university owns it, and I'll probably get $2, $3 for it eventually. What about resistance? That's always a very serious problem. So we looked at our organism, our TET-R organism with minocycline, and remember it had a MIC of 18. And you can see that over 15 days, which is the extent of this experiment, and TET-R reproduces about every 23 minutes, so this is 1,000 generations. You can see that we had 40% resistance after 15 days, and you can see that we're still almost within experimental error of the baseline with the hydrophile. Likewise, when we look at toxicity rates or survival rates of, this is human embryonic kidney, so this is a human mammalian cell. HeLa is a cancer cell line that's often studied for toxicity as well. COS7 is monkey kidney, also a mammalian cell line. And I think you can see that our hydrophiles really do very well in terms of toxicity. So this is a very encouraging result for us. Now, I want to conclude by talking about some slightly different compounds which are based on what would be tryptophan if it actually had atoms in it, and I apologize for that. Dr. Kim didn't take all of these, so I don't know what happened to them. This is the structure of the KCSA voltage-gated channel, the Nobel Prize-winning structure. And notice that tryptophan occurs only at the boundaries of the membrane. It's a striking result. Now, tryptophan is my favorite amino acid. I know you probably have other favorites, but I like tryptophan. And that turned out to be a good choice because there's actually an International Tryptophan Research Society that I was once invited to speak at. I didn't even know it existed. But tryptophan, even though it's the least common amino acid in proteins, is one of the most important. It's, of course, fluorescent, but it also is used as a stabilizer in many, many different proteins. So we asked, could we use tryptophan as a boundary for other kinds of amphiphiles, and would we get any biological activity? So it's easy enough to try. We put a spacer between 2 tryptophan molecules, and we've looked at arenes here and heteroarenes and alkyl chains and so on. And we've looked at the other 19 genetically encoded amino acids, and they don't do anything, only tryptophan does. So this turned out to be fairly interesting. These are a few of the compounds we looked at. And you can see that we get pretty substantial recovery of tetracycline activity from our resistant strain, and mostly we have pretty good survival. I mean, here it's less, of course, but mostly the survival is pretty good. So these compounds are currently under examination in our lab, and in fact, while I was here, I got notification that we're getting a patent on these. So if this were tryptophan, you could recognize it, and if you could see the atoms, that would be norfloxacin. We did what's called a checkerboard analysis, and I won't really go into the details. If you don't know what they are, don't worry about it. They're kind of hard to read and understand. But what's important here is that we compared our bis-tryptophan with norfloxacin and used it as an adjuvant combination, and we enhanced the potency of norfloxacin by 128-fold. So these compounds are remarkably potent. I would tell you exactly how they operate, but I don't know yet. We have had good success with them, and we're making other derivatives, but that work is presumably going on while I'm here. So a remarkable number of compounds can exhibit this kind of adjuvant activity and have good resistance to resistance, if you will, can increase potency, can be drugs in their own right, and they can have a wide range of structures and can have fundamentally different characteristics chemically from those that we had originally imagined. And so we can reverse antibiotic resistance, and we're hoping that will be a contribution to health. These are a few of the people who've worked with me on this project, and of course you don't recognize their names for the most part, but this was the best picture I had of the team at the moment, and I want to thank all of them for their wonderful effort. We've been supported by lots of organizations over the years, and I am extremely grateful whenever I get a grant, and I just want to thank you for a wonderful enrichment experience for me. Thank you for your attention.
So we're going to have a couple of questions. Oh, now I can see the audience. Okay. Questions? No questions.
I have some questions.
Yeah. Oh, sorry. Did not see you. Can we? Yeah, right there. Can you just stand up so they can give you a microphone? He has a microphone? No? Okay.
Thank you very much for the interesting talk. Actually, I'm asking as a mathematician out of my curiosity. So I look at the slides and everything. I just want to know, like, from your side, how you quantify the efficiency of that transportation, and also how you quantify the level of toxicity for different kind of structures. Is there any reference?
Excuse me, how we quantify
the efficiency of the transportation for each different structure of drugs that you are transported into the cell, and also the level of toxicity. Do you have some reference, or is completely empirical?
It's empirical. We're only looking at the improved potencies by MIC. We do have some PK/PD data that would be difficult to go into at the moment, but a couple of the things that we know that are relevant to this are that we don't have activity generally against the cytochromes, so those are not inhibited. And most people, when they think of a synthetic channel, they think of HERG and think that must be toxicity, but we don't have bad toxicity against HERG. So we have some PK/PD, but everything I've presented here and mostly what we do in vitro, gives us just the empirical numbers.
Thank you very much.
Thank you for the wonderful talk.
Thank you.
I have a question regarding the, yesterday or 2 days ago, we had a lecture about perforins and some other biological molecules that can create the pores inside the membrane, such as defensins, like proteins. Have you considered using as a positive control some biological proteins that can make a pore and compare the efficiency of the known antibiotic and your system? Thank you.
I don't think we're really at that stage yet. That's a very good suggestion, but we're still surveying. We have a bunch of new molecules, and we're still surveying for activity. And since we have, I haven't shown you, but we have some compounds that are about 15 nanomolar now. And we're focusing on trying to develop those rather than using our resources for comparisons like that. But it would be certainly something we'll need to do in due course.
Thank you very much for your wonderful talk.
Thank you.
I am wondering which is your perspective about the growing antibiotic resistance. Do you think with the great chemistry that you are doing, we have some hope that we will not reach a stage where the infections will not be treatable anymore?
Well, there already are lots of infections that are not treatable. Resistant infections in, I think, 2018 cost 35,000 lives in the United States. I think there were about 2.8 million cases of almost untreatable, and turned out 35,000 were. I think, based on what I know about emerging technology, there are a number of new antibiotics that are coming out now because both the CDC and the World Health Organization have published urgent missives about this. And there was actually a head of state meeting a year ago sponsored by the World Health Organization. Even the military is asking for proposals on this because, of course, they have people stationed around the world and subject to those things. So I'm always hopeful about technology. I won't say we'll win, but I hope so, or we'll be dead.
Okay, so let's thank our speaker for a great talk this noon. And you know, we have a lot of students in the audience, and you guys have to scan before the second lecture. So first,